kind of protein over another. So administration of the antisense oligo, as expected, blocked the late LTP in the wild-type mice. But it failed to block the LTP in the protein kinase M-zeta knockout mice. So this contrasts with the results of the ZIP experiments that Volk conducted, in which the less specific ZIP knocked down the late LTP in both wild-type and protein kinase M-zeta mice. So since Tsoukas was able to knock down late LTP by specifically disrupting protein kinase M-zeta mRNA translation in the wild-type mice, and the intervention was very specific for protein kinase M-zeta messenger RNA, and it failed to work in the protein kinase M-zeta knockout mice, well, after 3 years of debate, protein kinase M-zeta was back in the game as the mechanism underlying persistent and enduring LTP and memory. But Tsoukas and collaborators weren't quite done yet. They reasoned that the protein kinase M-zeta knockout mice must upregulate another kinase to replace the function of the missing protein kinase M-zeta. They examined the complete family of protein kinase C isoforms, shown again at the right of the slide. Remember these? We have the conventional, the novel, the atypical isoforms. Tsoukas and colleagues used phosphospecific Western blots, that is, protein assays that recognize the phosphorylated forms of the proteins, to see if the activated versions of any of these isoforms were upregulated in the hippocampus 3 hours after tetanic stimulation. They found that protein kinase M-zeta was upregulated in the wild-type mice 3 hours after tetany in comparisons with the levels in the unstimulated mice, as you would expect if protein kinase M-zeta is the memory molecule. In the protein kinase M-zeta knockout mice, of course, protein kinase M-zeta was just absent. Then when checking expression of the atypical isoforms, protein kinase C, iota, and lambda, they found that they were not upregulated in the hippocampus of wild-type mice 3 hours after stimulation. However, these isoforms were upregulated 3 hours after tetanic stimulation in the hippocampi of the protein kinase M-zeta knockout mice. This suggests that there's some form of biological re-regulation. If the protein kinase M-zeta is removed by knocking it out, there's a compensatory change in the functioning of protein kinase C iota lambda, which would explain Volk's finding that late LTP and long-term memory endures in protein kinase M-zeta knockout mice. It would also explain why ZIP would knock down late LTP in the protein kinase M-zeta knockout mice, because ZIP, originally thought to be quite selective for protein kinase M-zeta, actually has significant inhibitory actions on the atypical protein kinase C iota lambda. Still, Tsoukas took the experiment one step further. They used a drug that quite selectively inhibits, or somewhat selectively inhibits, protein kinase C iota lambda, called ICAP. and treated wild-type and protein kinase M-zeta knockout mice in late LTP and in a Morris water maze. Let's take a look at these experiments. In the top right graph, you can see that ICAP administration 3 hours after tetanic stimulation blocked late LTP in the in the protein kinase M-zeta knockout mice, really driving home the message that the protein kinase M-zeta knockout mice were using protein kinase C iota lambda to compensate for the loss of protein kinase M-zeta. In contrast, the wild-type mice, with their intact signaling through protein kinase M-zeta, were unaffected by ICAP administration after tetanic stimulation. And so then let's take a look at the Morris water maze. In fact, they used a modified Morris water maze in which on the first day, they allowed the mice to explore the Morris water maze, and they found that they all, both the wild type and the protein kinase M-zeta knockout mice, all swam all around the maze at baseline, as shown over here. Then, in the training, they actually shocked the mice whenever they entered the target quadrant. On the third day of this aversion training, you can see that both the wild type and protein kinase M-zeta knockout mice exhibited a robust aversion to the shock-associated target quadrant. But then they treated the mice in both groups with ICAP, the protein kinase C iota lambda inhibitor, prior to a retention test. And that retention test was 1 day later with no shock. And you can see that the ICAP treated wild type mice. The wild type mice nicely remembered to avoid the shock associated compartment or shock associated quadrant, demonstrating that interfering with PKC iota lambda long after the learning in these mice that do have protein kinase M-zeta does not affect retention. However, the ICAP treated protein kinase M-zeta knockout mice show a profound impairment in the recollection of the shock contingency, and they readily go out and explore the shocked quadrant. So to summarize, the protein kinase M-zeta knockout mice clearly overexpress activated protein kinase iota lambda 3 hours after tetanic stimulation, whereas the wild type do not. The protein kinase M-zeta knockout mice clearly use that overexpression of activated protein kinase C iota lambda to compensate for the lack of protein kinase M-zeta in terms of both late LTP and long-term memory. And in contrast, the wild-type mice clearly depend upon protein kinase M-zeta for the late LTP and long-term memory. They have no enduring activation of protein kinase C iota lambda, and interfering with their protein kinase C iota lambda does not disrupt late LTP or memory. So this double dissociation is really extremely compelling evidence that protein kinase M-zeta functions as an essential memory molecule responsible for long-lasting potentiation and memory in mice. And let's follow up on this by doing a little comparative neuroscience. If you remember way back at the beginning of the course, I described that evolution suggests that learning and memory mechanisms in the nervous systems of simple creatures may be closely similar to learning and memory mechanisms in the brains of complex animals like us. But I also said that there may be differences, especially as the complex animals may evolve more complex mechanisms to serve more complex problems. And remember, our friend the C. elegans that showed evidence for non-associative learning that could last for maybe hours, and the aplysia that showed evidence for both non-associative and dissociative learning with memory that could last for hours or even under extreme conditions, days. Remember those simple creatures? Okay, well, let's explore the potential that there are similarities and differences in our memory molecules. First of all, there is no evidence of protein kinase M-zeta or anything really quite like it in either C. elegans or aplysia. So it seems possible that in evolution, more complex nervous systems have evolved this kind of unique, self-perpetuating memory molecule. But then there's also no evidence that these simple worms and sea snails can form memories that last a lifetime. So maybe they don't need a molecule like that, not in their evolutionary niche. However, the aplysia do synthesize one representative of each of the three main families of protein kinase C isoforms, the conventional, novel, and atypical, or they're known as aplysia protein kinase C1, aplysia protein kinase C2, and aplysia protein kinase C3. And it makes all three of these in its neural tissue. And all three of the aplysia protein kinase Cs can be cleaved by calpain, pictured in the graphic at the upper right, to permanently remove the regulatory subunit and form kinases that, whereas they're not self-perpetuating like protein kinase M-zeta, they do have very prolonged activity, perhaps a little bit like protein kinase M-zeta, just without the ability to replace itself with this ongoing active form in the face of molecular turnover. And so, in fact, Glanzman's group showed that ZIP and chelerithrine erase long-term sensitization of siphon withdrawal reflexes when these inhibitors are injected up to a week after training in aplysia. The graph at the bottom right of the slide shows an experiment they did where aplysia gill siphon withdrawal was sensitized with a protocol that produced long-term sensitization of withdrawal that lasted 48 hours or more. In animals trained and treated with ZIP after 24 hours, a test at 24 hours showed no sensitization. The sensitization was removed by administration of the protein kinase inhibitor 24 hours after training. But if a scrambled ZIP was administered, a form of ZIP that would not inhibit the protein kinase, the sensitization endured. So it looks like the enduring form of the sensitization, although not mediated by the exact same mechanism as in the mammals, was some form of at least somewhat close approximation, where an ongoing form of activated protein kinase accounts for the late stage of sensitization in this simple sea snail. And there's one more thing I feel I really need to point out about protein kinase M-zeta. Work from Todd Sachter's group and others has shown that protein kinase M-zeta and protein kinase C iota lambda are found in association with neurofibrillary tangles in medial temporal lobe structures in Alzheimer's disease. I don't think we really know what this means yet, but we do know that the neurofibrillary tangles are formed by twisted clumps of hyperphosphorylated tangles of tau proteins that under normal conditions provide support to microtubule structures. It feels to me like a very powerful coincidence that a very enduring and ongoing agent of phosphorylation is associated with these hyperphosphorylated tangles. So as we learn more about Alzheimer's disease, we're coming to know that this is a much more complex disorder than originally thought. And it's shocking to think how little we actually know about factors that underlie vulnerability, etiology, and pathological consequences of the progression of the disease. However, one thing is certain. We need to know much more about the function of these biochemical and molecular signals if we're going to get a handle on what starts the process, what leads to the formation of the tau tangles in Alzheimer's disease, and how is memory actually being so profoundly impaired in Alzheimer's disease when we can see that a molecule like protein kinase M-zeta is caught up in these hyperphosphorylated neurofibrillary tangles.